educational satellite television programs.
Hello, teacher. Hello, students. Welcome to today's lesson on the basic techniques of a microscope. In our last lesson, we learned about magnification and resolution in a microscope. We distinguished the difference between the two and compared the resolving power of light and electron microscopes. Today, we will learn about a few basic techniques of using a microscope. We will discuss the steps involved in mounting, staining, and observing slides. Are you ready to begin, students? Observing specimen through microscopes requires skill and practice. It is important to understand a few basic techniques of preparing the specimen, using the microscope, and observing the image to ensure that you are getting the best results. Different specimens need to be prepared in different ways. Also, the preparation material differs between a light microscope and the electron microscope. The first step of preparing a slide of a specimen is mounting. Mounting means that a slide is prepared and the object is simply placed on the slide. A cover slip may be placed on top to protect the specimen and the microscope's objective. As well, to keep the specimen still and pressed flat. Let us go through the steps of preparing a mount slide. You begin by using a flat glass slide. Place the specimen on top of the slide with forceps. Add 1 drop of water to the specimen. If too much water is added, the cover slip will float on top, making it difficult to observe the specimen. Make sure the specimen and the drop go on the center portion of the slide. Ensure that the slide is not tilted while you are doing this. It is very important to keep the slide balanced and the specimen in the center. Using your free hand, carefully pick up a cover slip. Grasp the cover slip by the outer edges. Place the cover slip on top of the slide, making sure the edges of the cover slip match up with the edges of the slide. Do not press down on the cover slip. For the last step, continue to grasp the slide and cover slip by the outer edges, keeping the slide as horizontal and steady as possible. Place the slide on the stage of your microscope and get ready to observe. If the specimen requires staining to be observed, it would happen before or after mounting. Students, do you remember what the process of staining is? With a partner, take a few minutes to discuss how staining affects cells that are viewed under a microscope.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello, everyone. I hope that was a good opportunity for you to review what we learned in our previous lesson. Staining is the process of adding chemicals to slide tissue to make the cells easier to see. When stains are added to the specimen, they make cells or particular parts of the cell clearly visible. Cells that are stained are dead. These cells have been fixed or treated with chemicals so they do not decay. For light microscopes, only non-living materials need fixation, while living materials are not fixed. For electron microscopes, living or non-living materials are always fixed. The process of staining cells may involve immersing the sample before or after fixation or mounting in a dye solution. The sample is then rinsed and observed under a microscope. Some dyes require the use of a mordant, which is a chemical compound. A mordant reacts with the stain to form an insoluble color solid that is found in or on the sample. The mordant stain will remain in or on the sample when excess dye is washed away. For light microscopes, staining must be done using colored dyes to reflect light. For electron microscopes, metals such as lead and uranium are used to reflect electrons. The metals are used to scatter imaging electrons, therefore giving contrast between different structures. Typically, thin slices of the specimen are stained for several minutes with a water-based or alcoholic solution of the metals. Students, let us do an activity. List the steps involved in mounting a slide sample.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Hello again, everyone. How did you do? Let us go through the steps together. First, we use a flat glass slide that is free from dust or particles. Second, a thin sample of the specimen is gathered. Third, the specimen is placed on the center of the slide using forceps. A drop of water is added to the specimen. Fourth, a slip cover is placed on the top of the slide, ensuring the edges of the slide match up with the edges of the slip cover. Fifth, the slide is placed on the stage of the microscope, ready to be observed. Were you able to list all of the steps? If so, well done, students. The last technique we will be discussing today is observation. When observing specimens under a microscope, it is useful to make labeled drawings of what the specimen looks like. When drawing specimens, use a pencil. With a pencil, you can erase any mistakes you make, and you can shade in areas to show the different colors or textures. The specimen should be drawn in a circle. The circle represents the viewing circle as seen through the eyepiece. The specimen should be drawn to scale. This means that if the specimen takes up the whole viewing field, it is important that the drawing reflects this. All drawings should include clear and proper labels. Drawings should be labeled with the specimen name and the magnification. The labels should be written on the outside of the circle. This brings us to the end of our lesson. Today, we learned a few of the basic techniques for using a microscope. We discussed the techniques of mounting, staining, and observing. Until next time, thank you, teacher. Thank you, students.